So any questions from yesterday? No questions? Huh? Okay, so I'll be asking questions. Since you don't ask, I'll ask. Okay, so the plan for today is, I thought I'll solve a bunch of papers for you on adaptation, asexual adaptation. So this might be a little fast for some, but I'm hoping that at least we'll get some messages out. Okay, and I'll also explain things verbally and a slight bit of math. The reason I'm doing this is because I want to show you how we yesterday we learned some small models, single locus, only selection, only this and that. So how those little building blocks, how you can put together and learn something little bit complicated. So I'll give you the references later. And then I want to sort of compare the answers that we get from a Lenski experiment. Okay, so that's the sort of goal. Set up the theoretical model and then try to see whether or how it matches with this experiment on bacterial colony, bacterial adaptation, which has been going on since, anybody knows? 88, okay. Everybody knows the Lenski experiment, the so-called Lenski experiment? Okay, so I'll see that later on. Okay, there's a couple of things. So some of you asked me some questions which were actually very relevant, but you asked me after the class. So I think somebody asked linkage disequilibrium. So he was saying that what is this equilibrium in that? So you remember we said yesterday disequilibrium, linkage disequilibrium. So is this equilibrium same as that stability analysis wala equilibrium? As far as I know, nothing to do with it. Okay, so don't get confused. But I wish he had asked this question because maybe others also had the same confusion. Others have asked for references. I'll send it by this evening. Okay? Any other question or something which everybody should know? Oh? Yeah. No, so I think, I don't know why this term was given. This was in Levantine and all. They gave it in 1950s or 60s, something like that. And I think there's some reason for it. So I think Slatkin has a Nature Reviews. I forgot what the motivation was, but if you want to understand a bit more, you might want to look at this. So it's on LD by Slatkin. I forgot the year. Yeah. Yeah, so there was this linkage equilibrium when D is 0. So in that sense, yeah. Not in equilibrium. Yeah. But it's not the same. It's not like that, the sort of equilibrium things that we were doing in the morning. Okay, so dt is not equal to, it becomes 0. That's all. Okay, so let me start with this. So I'll follow some of these papers for today. So there's this guy, Alan Orr. I like his articles very much and I highly recommend them. Very readable. So this is in genetics. So as I go along, I'll mention others. So this is one basic paper and experimental paper is this one from Lenski group. Okay, so we'll see that. Okay, so I want to talk to you about asexual adaptation. So now today there won't be any single locus and all that. There will be mutation, drift, and selection. No migration. That's the only difference, okay? So but it's a more full-fledged model, more general model. Okay, so we'll see how it goes. So I'm thinking of an asexual population of size n. And it's a large population. So what is meant by large, we'll see later. So that's a sort of important word, however. Okay? So I'm saying it's about adaptation. So what I'm thinking is that there's a population of asexuals, bacteria, whatever you like, okay? But this population is initially well adapted to a particular environment. And then suddenly you change the environment. So for example, in Lenski experiment, you just give it less glucose. So suddenly it has become maladapted. And the question is, now after that, I'm not going to change the environment anymore. And in this new environment, this population has to get new mutations which are hopefully beneficial and increase its fitness, right? Okay. So then it will accumulate mutations, beneficial mutations. with time. Okay, and we want to see how that behaves. Okay, so this is one result I'll give you. It's an assumption right now that the average fitness grows like e to the power capital RT. Again, this R has nothing to do with Vishnu's R. This R is the speed of adaptation. Why? Because if I take log of this, okay, so this divided by T, or let's write it like this, equals R times T. So if I show you something like this, log W bar by T, when it follows from here, right? That's okay. But this is the speed. R is the speed or the rate. Why? Because you told me speed is equal to distance upon time. But this distance is in the fitness space. It's not the real space distance, okay? So the distance in the sense of how fitness has changed over some period of time, okay? So that's the speed of adaptation. So there's a lot of work, physicists, biologists, and a lot of people to calculate this object. And then you see that in experiments, so that's why there's a motivation to understand it or calculate it. Okay, so this is our definition. So W bar here is the Wrightian fitness. Okay. So please keep this in mind. This is the object of interest. So before I start any calculation or anything, let's figure out sort of qualitatively what we expect. So I've already introduced N. So what do you think? Would the speed of evolution or adaptation, would it be large in a larger population or larger in a smaller population? How do you think it depends upon N? What's your sort of, simple expectation? So what's going on? So you have this population of size N, and then it will have these each individual. So let me introduce this. So I said beneficial mutations have to come. Is that right? Beneficial mutations are the ones which will drive the adaptation, right? That's what it means, right? So they occur at a rate UB. This is standard notation. So UB is the probability that an individual acquires a beneficial mutation per unit time. Per unit time. Okay? So each individual might acquire in this one generation. It might, right? May or may not. So remember yesterday what we said? So you look at each individual. It maybe got a mutation or not. You put a 0 if it didn't get. Maybe you got it or not. If it got it, you got to put a 1 and so on. And you count the total number of mutations in that colony of size N. How many average number of mutations do you expect? Is the question clear? I'm asking, I have this N individuals, 1, 2, 3 and so on, N individuals, okay? Let's put a number, 3, 4, 5 individuals, right? So maybe it got a mutation, good mutation. If it got with a probability UB, maybe this didn't get, this didn't get, this got, this didn't get, right? Reminds you of something that we did on the first day? Toss of a coin, very good, right? Heads, tail, tail, head, tail. So I'm asking, this is the thing. Monday I told you, there are something which will come again and again and again. So you should never forget it. Okay, it should be part of your bloodstream or something like that. Okay, so now I'm asking, there will be distribution of these beneficial mutations, the number of mutations a population of size N gets. What's the average number? N times? Yeah, very good. Yeah, I'm impressed. Right? So this is the average number of beneficial mutations per unit time. Right? Okay, so what do you think? Now looking at this formula. So if I have more mutations per unit time, there will be more speed, more faster evolution, right? So this little 1 minute formula tells you what I think most of you already said. That is, that if the population is large, the rate of adaptation would be larger, right? Because more guys to get mutations, right? Okay, but this 1 minute calculation tells you that this rate of adaptation is increasing linearly with n. Okay? So you expect R to be proportional to n, linear in n. Okay? So we're just doing some simple 1 minute thing, calculation here right now. So is this okay with everyone? So I wanted to ask you how the speed increases with n, and this simple thing tells me it should increase linearly with n. Okay, so this is okay for now. But what happens is the following. In asexual populations, if I have too many mutations, so UB, let's say, fix it, but the population is large, then there will be lots of mutations per unit time. So I'll draw sort of allele frequency trajectories to explain what I'm getting at. So suppose I have this good mutation, and I'm plotting allele frequencies of various sorts. Okay, so there's allele frequency of one type, it's a good one, and it started to increase. So this time, started to increase and became 1. So that's the sort of a scenario which will, you expect, if the fixation happens, right? But while this is going, so this is 1 frequency. While this is going to fixation, what might happen is that another one might come. Let me draw it better because I want to make some points here. So while this is going towards 1, another beneficial mutation might come, and what do you think will happen? So I have another beneficial mutation. This increases the fitness by an amount larger than this one. So when this beneficial mutation occurred, it increased the fitness by, let's say, 0.01. And this increased the fitness by 0.1. What do you think will happen now? And there are lots of things which are wrong with this curve. And by now you should be able to tell me. Let's forget the pink one for a second. What's wrong with the blue curve? So I said beneficial mutation came. So there was this big colony of size n, million particles, individuals, and this one good mutation came and tuck, 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 marched up. Is it? What's missing? Forget the pink curve for a second. It just marches up. Drift, yeah, loudly. Yeah, otherwise, I feel that, I haven't taught properly. So, yeah, so okay, I have taught okay, I guess. Yeah, so it will march up provided it did not get lost due to drift, right? Okay, so it can march up if not lost, okay? That's one thing. But let me grant that, let's say it didn't get lost, so it will climb. So this climbing, do you think how much time would it take? Suppose the pink one is not there, the blue one, how much time does it take for it to reach nearly 1? Nearly 1. No, that's not a time. Time. How much is it? Look at your notes. I need it. How much time does it take? Just tell me intuitively. Huh? Log n by s, right? So this will take a time to climb up of time. The blue to be log n. Let's call it blue fitness, okay? This is blue fitness, right? So it will climb like this, but this has a higher fitness. So that's why I made this more steeper. Is that okay? Is that what you would have drawn? So this guy is growing like e to the power 0.01t, and this is growing like e to the power 0.1t. You remember yesterday what we did? Logistic equation. At short times, you can forget this factor, which means x goes like, right? Okay? So this has an s 0.01, and this s has an s 0.1, sb, right? So will it grow faster or slower than the blue one? Faster, right? So therefore it's okay if I plot it steeper. Okay. So then the effect that happens is if there are too many beneficial mutations, then while this is going to fixation, a better one might come along, and now these two will compete. So if there's a competition, which one do you think will survive? Which one? 0.1, the pink one, right? Okay, so this guy goes down, This one also, provided it didn't get lost due to drift, this goes up. Okay, so what has happened? And now you can sort of generalize this argument. If the pink one, if there's something like this S, let's say 0.2, it would have come even more steeper and not allow this one to fix, right? So there is a competition between beneficial mutations. In both sexual populations this happens, as well as asexual populations. If we ask about this effect, I'll just write it down. This is interference by beneficial mutations. It happens in sexual populations. I'm assuming asexual one, but there's a classic paper. If you're considering sexual populations, we recognized earlier, before here, for sexual population, it's called Hill-Robertson effect. And asexual populations, it's called, anybody wants to tell me? No? Clonal interference. Ask me a question if you have. Oh, minus x I removed for the logistic because it's short times is good. Okay? Okay? So this competition, so this is because now what I'm thinking of is not a single locus model. I'm thinking of a genome which has some finite length, or in fact infinite length, very, very long genome. This is what I'm going to assume, because you know, if I'm standing here and there's a wall here and I'm going to paste only in this part, and I don't care whether the wall is over here or in Banerghatta, right? Right? So similarly, I'm going to think of a genome of an infinite length. Okay, so that's what I mean by infinite things. And this genome in which lots of things are linked together, this kind of effect will be very strong. Whereas if I have a sexual population, we learned yesterday about linkage disequilibrium, these associations will keep breaking. There is this kind of effect, but it's much milder. Okay, so let me just go back to asexual populations. So what is this effect doing to the rate of adaptation? So this guy was marching up, and therefore you could have stepped up, you could have increased your fitness. But because a better one came along, which is good in some ways, there was a loss of beneficial mutation. It's a waste of beneficial mutation, right? So what do you think? Would the speed be same, increased, or decreased if I have clonal interference? So I have a larger population. Everything else is same. So I have population size million, and now I have a population of size billion. And because beneficial mutations are coming at a rate n times UB, there are more beneficial mutations coming in this large population, and they'll compete, and you know, one of them might win. So which one do you think is going to adapt faster? Smaller population or the larger population? With the one, so you expect that it will have less, the one which has less interference. That is, if I just increase the population size, my naive little one line calculation tells me, have more population, you will have more beneficial mutations in this room, right? Let me just pack up all these smart guys, right? So I will have lots of papers and stuff like that. But then you will start competing, right? So therefore less papers might come. I mean, sort of something like that, right? So you expect, therefore, that the rate of adaptation would slow down. It will not increase linearly at large enough populations. So it's not going to do this. At some value, it might actually do, okay, let's do this only. When I take the clonal interference into account. Is the effect clear? Okay, this is a simple physical effect. Is it okay with everyone? Yeah. Yeah, that's what. So it's not obvious. It's not so obvious, right? Because you will say, yeah, so fine, 0.01 guy, right? This is 0.1, so therefore there's a big step up, right? But so would it increase, or is it that I could have fixed 0.01 and 0.1 and so on? And would that have increased the fit? See, 0.1 is a little extreme, so I shouldn't have taken, I just took it like, you know, Generally, you don't have such values, so maybe that's why you're getting this impression. Suppose this is 0.015. Typically, numbers are sort of similar-ish. They're slightly better, not very much better. I'm saying the rate of increase, the speed would increase. Speed would increase with n. The speed would increase with n, but not as fast as n. So, good question. So, I didn't say it very clearly. If I have a larger population, then the speed would be higher. So, the speed of in a billion is higher than the speed in a million. Okay, so I think I said wrong. So, speed in a billion would be higher than the speed in a million population, but it will not increase 1,000 times. That's the point here. So, the speed would increase with larger n, but not as fast as linearly. So, this is the linear growth, right? Because more mutations are coming, n times p binomial. But now I'm saying I expect on a basis of verbal argument that it may not increase as fast as linearly. So this is what we expect on a basis of a verbal argument. This is very important effects. These Hill-Robertson effects and clonal interference, they're very, very important. So if it's not clear, ask me. Is this okay? Yeah.
Doubt. So let's say right now here you have given an example, like firstly one type of variant was increasing in frequency, then the other one came which has better fitness, so it will take over. But this analogy can be extrapolated. Then when this 0.015 is increasing, then some other one will come. So in that manner, if we compare both of them, because there the size was small, so that will reach frequency 1 at an earlier time than this one. So means this will get
this will, although it's coming later.
No, so in this model, it will take more time to reach frequency 1.
This one, 0.015.
No, so it will increase, but then some other mutation will come.
Better.
Yeah, so then that will keep on increasing. Then when it's about to reach, then again some other mutation can come.
Yeah.
So if we compare small population, large population and a very, very large population, so in a very, very large population, the speed will be slow. Means it will never reach 1. Yeah.
Speed will not reach 1, yeah.
No, not the speed, the frequency will not reach.
But I mean, there's a, so, but do you think the SB is infinite? I mean, SB, I already said, but do you think the fitness effects are, there's a limit to them? No. So, I mean, it's some ways, so I have to, introduce some, so I think DFEs were already introduced. The distribution of beneficial fitnesses, they're such that, so this is a general character, large fitnesses are less probable. Okay, so I'm coming to that in a second. I just right now keep it verbal. I think later on you'll understand better. Is this okay so far? So the fitness, the rate would increase. The speed of adaptation would increase with n, but not linearly at large enough population. Okay. What do you think about, so this is one thing. Okay, yeah. So this is one parameter n. How speed might depend on n. What about the speed with mutation rate? How does it change with mutation rate? What's the expectation? Are there only beneficial mutations? What are the types of beneficial mutations? Oh, sorry, what are the types of mutations? The beneficial mutations and deleterious and okay. Okay, we'll just say neutral, beneficial and deleterious, right? These three types, right? So neutral I'm going to neglect completely because there's no effect on speed, right? So we're checking what? Change of the fitness. Fitness I won't change, right? So I'm going to ignore it completely. So I spoke about beneficial mutations. What about deleterious mutations? They should also do something to it, no? So there's a UD as well as a UB. So we have a UD, which is the deleterious mutation rate, just like we had a UB, which is the beneficial one. And experiments, and I'm not sure from where this comes, but people generally think that this ratio is fixed for a particular organism. The fraction of beneficial mutations or sites, they remain fixed, okay? So the total mutation rate is UD plus UB. So I'm now asking you the following. How do you think R changes with total mutation rate? So let me simplify this a bit. Might be easier to think. UB is so if I fix PB, it's the same almost as UD. So if I have very large mutation rate, what will happen? What do you think will happen? If it's confusing you, forget that. You just use your intuition or whatever. If the mutation rate is very high, what is more important, UD or UB? UD? Why? No, why? Why is UD more important? If the total mutation rate is increased, why doesn't UB increasing?
When the mutation rates were low, even if there were a few deleterious, they were selected again, and by the fact that they would by chance, be good at.
No, right now not just to check about chance, just the dependence on, suppose mutation rate is 0. What's the speed of adaptation? 0, right? So we know 1 point. Let's now increase the mutation rate a little. And the fraction of beneficial mutation is, let's say, 10 to the minus 3 or something. So I have this long genome, and only a tiny part of it has beneficial mutations. And total mutation rates is very tiny, 10 to the minus 7 or something like that. Would the rate be, it has to increase, right? It cannot remain 0, because I'm allowing beneficial, I'm allowing mutations. So would it increase? This much is okay with everyone? At very high mutation rates, what is the important thing? Beneficial mutation rate would increase, deleterious would increase, but what do you think is more important? Deleterious one. So therefore, the rate of adaptation what? So slowdown means what? How should I plot this? It will go like this. Is that okay? So this slowdown is happening because of deleterious mutations, and this increase due to beneficial mutations. When U is large, why delete? No, so there are, both of them are there. Actually, it turns out. So UB is generally very small. When the total mutation rate is large, the deleterious mutations have a higher effect. So right now, it's sort of you expect roughly, but I haven't shown you anything like that. It is what you expect. So therefore, you expect that perhaps rate of adaptation goes like this. You think so?
Small question.
Yeah.
So our U is UD plus UB. So we can achieve high U by having high UB also and having UD small. But this graph is saying no matter whenever U is high, your R is small.
Whenever U is high,
your R is small. So this means even if you are having a high rate of this beneficial mutation, still your R is.
Yeah, so at a very large one, it's the deleterious one. That will impede the adaptation. So the thing is that total mutation rate is high, right? So UB would become high, so does UD.
If we just look at this equation,
so let's take some numbers.
UD plus UB.
Sorry?
Fraction is less than 1.
Yeah, that's always true, right? So at higher mutation rate is basically UD. I think that's a good way of saying. So at large mutation rates, basically I can neglect UB. You can think that way. Somebody was asking, right? So at large U, I can basically neglect this one. So use approximately UD at large U. That's one way of seeing it. Yeah. PB is a constant. Yeah. PB is a constant. Yeah, that's true because beneficial mutations are rare. Most mutations are deleterious. Okay, is this roughly okay? Okay, so, okay. Okay, fine. Right now, let's just buy it. So therefore, you expect that the speed of adaptation is maximum at some optimum value. So then people talk about this, but it's not clear whether it's really useful. There's some optimal mutation rate at which population adapts the best. Okay, so this was still verbal, and some of you have questions, really, is it non-monotonic? Actually, it's not that obvious, because I know the answer, so maybe I sort of bought it. It could have been, I think it's legitimate to ask the question that you guys are asking. So maybe, U has some value, right? I mean, some biological range. Maybe the function could have been like this, right? Or it, at least you expect it to be, or maybe it could have been like this, right? Maybe decreases, or it could have just kept it increasing. That would have been also possible, right? So would you have argued for this verbally? You think verbally would have been okay, this one? It would have been okay, no? Oh? Yes or no? No? Why? Yeah. So he's saying that U is very, very large, that the rate of adaptation is very high. But that's just saying that mutation rates are the beneficial rates are only important ones. Somehow that implicitly assumes it. Okay, so this is at least confusing sometimes to me also, especially since there are words and all that. So I'm going to write down a slightly involved, not slightly involved, but it sort of follows on what we learned yesterday. Okay, so I'm going to take you through this calculation because it's pretty standard in the literature. Okay, and if it's confusing, it's okay. You can just have a look at it and look at the paper later on. But I hope you can at least get some idea about how these things are done. Okay. So I have UB and UD. So I would like to understand these curves a bit and hopefully come to Lenski experiment. Okay. So we have this, and there's this one, and there was a UD. Okay, and whenever a beneficial mutation occurs, it's such that it increases the fitness by a factor 1 plus SB. What does that mean? So if a mutation happened here, then the fitness became 1.1. So the beneficial fitness effect was 0.1, right? Similarly, if the mutation was deleterious, it decreased the fitness by a factor 1 minus ST. Okay? So whenever a mutation occurs, it doesn't change the fitness by the same amount. Right? So it could have, now we are not thinking of single locus or anything. Okay? So we have this long genome, and if mutation occurs here, maybe it increases the fitness by this amount. If it occurs here, maybe it increases fitness by this amount, or it decreases by some amount, and so on. Right? So these SB and SD are random variables. So what was this thing that you learned earlier? There's a distribution of these selection coefficients. You learned that already. What's that called? Distribution of fitness effects, DFE. So there is a distribution of S. If the mutation is neutral, then there is some weight, right? If the mutation is beneficial, then what do you expect? So do you think so there are lots of such mutations that increase the fitness by this amount. Are there more mutations which increase the fitness by this amount or less? Less. So could you draw something there? What do you think this part looks like? It should decrease. Very good. So you expect generally something like this. What about the other part? So this is the beneficial part. And this is the neutral part. So this is the wild type here. What does this look like? Decrease, increase. Decrease. Okay. All the way to 0. And this is minus infinity or some minus large value. So this is like 1. to some very large value. Does it decrease all the way to 0? How does it go? So if the deleterious effect is very, very bad, if this mutation occurs, it just kills me. What are those mutations called? Lethal mutations. So are they there or not? They're there. So there should be some weight for them, right? So this is the SD part. This is the SB part. And there is some finite. In fact, there is a lot of weight here. Okay, this is how typical DFE looks like. Okay, something here, something there, and there's a lot of weight in the lethal mutations. So these are the lethal mutations. Okay, so what I'm saying here is, when we're talking about clonal interference or something, and we're saying that mutation occurs and increases the fitness effect by SB or SD, what you mean is that perhaps something came from here or here and so on. That's what we have in mind. So I think somebody was asking this question about SB, SD, how much value, something like that, somebody was asking. So I have this distribution in mind. Okay, yeah. Why there's so many lethal mutations? Deleterious mutations generally are more, but specifically lethal ones are generally more. I don't know why. No, they are deleterious, but they're strongly deleterious. A lot of weight. So he's saying, why are lethal mutations more? Why are there, lots of lethal mutations? No, yeah, but there is quite a bit of weight. Typical DFEs have quite a bit of weight at lethal mutations. If they are more than this area or not, that I don't know at least. Okay, but there is it's not a teeny weight, it's a substantial weight at lethal. I don't know the biological reason. Okay, so what we have in mind is something like this, and I'm going to simplify this real DFE because that's where the calculation has been done previously. To write down at least a very simple expression. So I'm going to assume for today's talk that DFE is something like this and this. I'm going to ignore all this part. Okay? So I'm saying either lethal or some weight here. That's it. And over here. That's what I'm going to assume, because it simplifies some things. Okay, so let me just write the expression, then we'll see how it goes. So now, I'm going to write an expression first, and then we'll see how sleepy you are. So you told me already n times UB is okay with everyone, right? Okay, so because it's about fitness, every time a mutation occurs, let's say it increases the fitness by the same amount, SB. Okay? Is there anything else? So, let me say it again. I'm sorry, I think I'm not being very clear here. So let me step back. Simplest case. Let's ignore clonal interference for a second. How would allele frequencies look like? How will they behave? So this guy is somebody, and it comes and goes to fixation. Then another better one comes. And so how would it look? Suppose there is no clonal interference. How would the second allele frequency look? Which is better? Yeah. So it will come after this guy has reached. If there's no clonal interference, okay? There's no interference, so there's a low rate of beneficial mutations are coming. So while this guy has already spread, nothing is happening, nothing is happening, nothing is happening. Then a new one comes, and then it spreads. Quiet, 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 like a punctuated evolution. Then again another one comes, and so on. This is the sort of very simple model that people have analyzed in great detail. It's called periodic selection. Or sequential fixation. So what this is saying is that the time taken between two fixation events is nicely well separated. And in that case, it's very easy to write down the speed of adaptation. So let me write it and then we'll see. So what do we want to do? A rate is, a speed is distance upon time. What's the distance traveled? Suppose this had a fitness, let me come to that in a second, but let me say simple. Here, this had a fitness 1 plus SB. This had a fitness 1 plus 2 SB. Then the third one came. That had a fitness 1 plus SB, 3 SB, and so on. So what is the distance being traveled in the fitness space? This SB. This SB, distance traveled per unit time, per fixation time. Sorry? Addition? No. Distance traveled, distance. Distance traveled is SB. Yeah, she's right. Right? So this is the distance traveled per unit fixation. But what's the time? So this has gone from 1 plus SB to 1 plus 2 SB, 1 plus 2 SB to 1 plus 3 SB. But what's this time scale I'm talking about? The time scale is this one. What is this time scale? Let's write it again. It's the time taken for a beneficial mutation to come. Beneficial mutation rate is NUB. So therefore time taken is, what is the difference between time and rate? Inverse. So, does it make sense? Let's forget clonal interference for a second. Distance upon time. Distance traveled SB and time taken is the time for mutation to come along. You think this formula is correct? In that scenario. Okay, I'm saying this is speed, distance as you said correctly, and time taken is what? Time per fixation. What's the time for fixation? It's this time that the mutation should come. The time for the mutation to come is inverse of the rate. Rate you told me, all of you, NUB in the beginning. So therefore time is inverse of that, right? So therefore this is the speed I will write. But this is not still correct. What is missing? So yeah, but it's average, this average time. It's that approximation, so good. It'll be different for each of them, but if I average it over, that's what I'm writing here. Okay, it's the average speed. What's missing? Okay, I'm only speaking, okay, I want to hear now from you guys. I have spoken how much? A lot. So enough of my talking. So what's missing? Distance upon time, speed. But now it's not just speed equal to distance upon time. You have to bring in population genetics into it. So combine mechanics that you learned in 10th standard and population genetics which you learned yesterday. So I'm going to take some rest. My speed is going to be 0 right now. Come on, chat with each other. It's okay. It's not an exam. So discuss with each other. If something is not clear what I'm asking you, ask me now. Is the question clear?
We are expecting how to change with the population size.
I want the full speed expression for speed, which has N and U and S, selection, mutation, drift. All three are there. Tell me, what's missing? This is no SB. SB is there. Without selection, how would neutral mutations don't make a change? So let's hear for some others. Time? Time is this. This is the point time. Back guys. Back guys, not bad guys. Back guys, anybody? Huh? Okay, I made a mess it looks like. Huh? Come on, tell me. Something is missing. And I know that you know, but you have to know that you know it. So do you know it? What's missing? See, it's not a point, there's no point in going forward unless we get this one, this point fixed. What's wrong with those curves? Type in. No, that was when we have interference. I'm saying now population size is not so large. This sequential fixation. So I said ignore CI, ignore clonal interference. It's the simplest thing that you can do. This is something, you know, I don't know whether I should call it basic. Yeah, actually basic, yes. What's missing? This is okay with everyone? Nothing is missing? Slope? Slope is speed, no? So, speed decreases when? No, but that is when I take clonal interference into account. I'm saying now, this guy comes, spreads, quiet. Another one comes after 2 days and spreads. Third one comes and spreads. So there is no competition. Let's have some ideas here, speculations. For each row, I want a speculation. Last row, let's start with you. What's missing? Selection, mutation, and drift, something is missing. What? Give me just some random idea. Initial point when the new mutation comes. So I'm saying that's typically 1 by NU. So it's all on the basis of averages. I'm not doing a very nice calculation. It's on the basis of averages. So everything is typical value. Okay, third row. Any speculation? Except Archana. Yes, we haven't taken details. Yeah, good. That's one thing. Yeah. So where are the deleterious mutation? Right, that's true. But something even more basic. So second row. Back mutation? Back mutation. Yes, but they are quite rare. So let's forget them. Some other speculation. No, this is asex. This is not a speculation. Come on. What's missing? First row. Total rate of mutation? So UD we ignored, no? So that she already said. That's a good point. Yeah. First row. Anybody? Fitness term is taken care of by the selection coefficient. That change in fitness for fixation is SB. Because the fitness is 1 plus 0.1, then it becomes 1 plus 0.2. So every fixation event, there was a 0.1 increase in the fitness. That's what the numerator is. Big clue. I already said it today also. I don't know. What does it mean? You have to say more. Take the mic so that everybody can hear. NBU is less than 1.
So here, population size has been considered. We're saying that there are n individuals and with some beneficial. So mutation comes around with this much of a rate, which is we said in UB. Now once that mutation comes, it has a rate of fixation, how strongly it is selected, which is SB. But even after that, there is, depending on the drift factor, it will take more or less time depending on the size, right? Like if it is a small population, might, even though it's a strongly selected.
No, so what do you think I should add? I mean, add, subtract, or multiply, divide, whatever it is. So
A 1 by n factor.
1 by n? Why?
Larger population, lower.
What does 1 by n give me? Huh? Yeah, it will cancel off. In fact, I will not get rate to be increasing with n. Okay. Yeah, it must be some probability because the theorist is talking. Okay. Come. Yeah, it's a probability. That's a good clue. Third row. What is that probability? Good. That's a good clue. Third row. What probability are we talking about? Sorry? SD we are ignoring. UD, SD, ignored. Probability. That's the clue. Huh? Huh? Probability of B? They're always better. I told you exactly 1 plus SB, 1 plus 2 SB. So this is like deterministic. I forgot that DFE also now. Now that we are, 2 steps forward, 1 small step backwards, I hope, probably. So let's do this much. Probability of drift? That's all in the selection coefficient, some effective selection coefficient. What is that? So, okay, yes, we have a jackpot event here. Yes, come on guys, how can you forget? I have spent my blood, sweat, tears telling you about fixation probabilities. How can you forget that? Yeah, that's what, fixation probability. How can we not talk about that? How can this guy just come? It's a rare mutant, right? That's what I said at the beginning. How can it just march up? I'm drawing the things that we are able to see. They are the lucky guys, good lucky guys, right? But what's the chance that they are the good lucky guys? They got fixed. The probability is fixation probability, and from Kimura's formula, if you simplify, which I haven't told you, it turns out to be 2 times the advantage. Okay, so yesterday I wrote a complicated formula for you. I said pi is right, but if this SB is small and NSB is large, that's where selection is, because otherwise neutral case, then it simplifies to 2SB. So this is also a very famous formula, even older than Kimura's formula. It's due to Haldane. (1930s). Okay, so we have to take into care of that object, right? So this is the rate of adaptation if we ignore deleterious mutations completely. Okay, so therefore, so what happened now? We said something about rate of adaptation being, according to this, it increases linearly with UB or U, right? Not non-monotonic, right? So, what's missing now? Something is missing, right? You guys told me already, deleterious mutations, right? So I assume deleterious mutation I'm going to totally ignore. So, what did we do? We started with a very simple thing. We say that, it just depends upon the number of the beneficial mutations that come in. That was this NUB factor. This was a distance traveled, and this is a drift factor. Okay, so this is mutation partially. This is the selection factor, and this is the drift factor. So I haven't done a good job of it yet because I haven't taken the deleterious mutations into account. So let me do that. So let's say that the deleterious mutations are such that whenever deleterious mutation occurs, is this okay with everyone now? At least this much. Okay? So let's say now that the deleterious mutation is such that its effect is very large. So whenever I have a deleterious mutation, because I can have beneficial and deleterious, beneficial, deleterious, and so on. Suppose deleterious mutation occurs and it decreases the fitness a lot. Let's take a sort of a very limiting case. Okay, so it's nearly lethal. Then in that case, what would happen? See, this is some question that you would ask yourself. No, you would say, okay, she's saying about mutations interfering when they are beneficial mutations, right? You think deleterious mutations can interfere too? Yeah, somebody say yes? Yeah, how? Can you take the mic so that everybody can hear? So we talked about interference of beneficial mutations. So deleterious mutations?
So in the beneficial mutation, what happened is because it was much more fitter in that sense, and it was going up, and the other one was going down. But because this deleterious mutation is much powerful than the beneficial mutation is what we are assuming, that will take the entire
No, hold on. I'm not asking for the competition between beneficial and deleterious. There, of course, beneficial will win. The competition between 2 deleterious mutations.
Yeah, so the one which is more
No beneficial mutations for a second.
Yeah, no,
Okay.
but one is more powerful than the other, right? That's
Yeah.
what you are saying. There is one deleterious mutation and there is another one.
Okay, yeah.
So the first one was dropping down the population basically, or the allele frequency was going down with it. And whatever rate it was going down, the rate will increase much more with the other one.
Okay. So what happens if I have two deleterious mutations, the effects of two, a very strong deleterious mutations? So which one would be forming the new generation? The stronger deleterious or weaker one?
Weaker
one, right?
Yeah.
So here I'm assuming that all deleterious mutations are very strong. So therefore, if an individual has, let's say 5 deleterious mutation, if it gets another deleterious mutation, because already population has 5 deleterious mutations, 6, 7, 8 and all will not spread. They will have 0 chance, or they just will not spread. Okay, so in that sense, there is an interference between deleterious mutations. Okay, so there's an interference, and this is again a strong effect for asexual populations. So here I have not derived the formula at all, so I'll just give you the answer. Okay, and it was introduced actually in a different context when we have neutral variants. So it has some name. It's called background selection. Where is it? So this is interference between deleterious mutations. There is now a general term that is used. Okay, so in this case, this has to change a bit, and let me just give you the answer. It turns out the population size does not remain n, it decreases. The population, because most mutations are going to be lethal. That's what I'm assuming here in this little model. So therefore, the population that's going to form the next generation would be somewhat smaller, right? So that effective population size, it turns out, is given by this formula. So this factor comes from some Poisson distribution, which I haven't talked to you about at all. So please, you have to buy it today. Okay, rest is same. This is UD by SD, sorry. UD to SP square. So this is the rate of adaptation or the speed of adaptation without taking clonal interference into account. SD would be pretty close to 1, yeah. Okay. So does this formula gel with this expectation for the rate of adaptation? So now I have taken into account what factors? There is deleterious mutations, beneficial mutations, deleterious effect, beneficial effects, and fixation. Drift. Right? So, does this look like a better model? Does it make sense? Just 1 second. Speed is increasing linearly with n, but how is it changing with the mutation rate? How is it changing? Yeah, Krishna. No, this one I have to introduce a full model. So this I'm just giving you the answer. It's an old result, 1973 or something like that. I'll give you the reference if you're interested. So could you please answer this question for me? How does this look now, the rate of adaptation with the mutation rate? Plot and tell me. Does it increase with the mutation rate? This expression, does it increase with mutation rate? Yes or no? Where are the mutation rates? Can you guys tell me? How does it look? Yes or no? Third row. Increases how? How does it increase with mutation rate? Yeah, but linearly, right? And decreases as exponentially. So, it's not like this, okay? It goes like this, according to this little calculation, right? So when we take in the deleterious mutations, beneficial mutations, deleterious effects, beneficial effects, drift into account, we get a simple expression for the speed of adaptation, which is increasing linearly with n and is a non-monotonic function of the mutation rate. But now you can see the increase was because of which term? UB. So therefore, that's what you guys said, no, there should be more beneficial mutation rate, so therefore rate of adaptation would be high. But the decrease is due to deleterious mutations, right? That's what you wrote out there. Is this okay, sort of? So I think we won't have time to do anything more about, clonal interference. I'll just give you the answer. So I won't go through the calculation. I'll just give you the answer. That you have to set up this model with the clonal interference in which things will come and compete, etc., etc. And the calculation is pretty simple in the sense of whatever we learned yesterday. If you know that much, you can basically follow the paper. Okay? So if you're interested, you can look it up. But let me just give you the answers. So it turns out that this rate instead of increasing linearly with n, here it's going like n at some populations, and then there is a change in the behavior. It increases logarithmically with n. So speed is increasing with the population size, but much slower after a while. After a while meaning, for a large enough population size, the rate of increase is very slow, is only logarithmic. No, it's just separate. You haven't included it. So this is due to clonal interference. Okay, so the reason I wanted to do this is, again, about what I said earlier, namely, we want to tease out what are the important things, right? So have I really done that? Okay, so there's a model, people have been thinking about it for theoretical reasons because people have been interested, especially physicists, in calculating speeds of all sorts of things, and there's a whole lot of things, not necessarily in biology. Okay, so there's a whole lot we know. Okay, so we just calculate it, fine, for the sake of it. But is it useful? So there is this experiment by Lenski. He has been doing this experiment for a very long time, like 88, somebody said, and he found the following in the experiment. So this is from the Pfizer paper. So I'm plotting W bar, not with respect to n or anything, it's with respect to time. Okay, so it looks something like this. It increases rapidly till about 5,000 generations or so, and then it's increasing very slowly. It's sort of in a log scale. So this is like 50,000. So this is, I think, about 1.4. Is some relative fitness. So if I show you this graph, what do you conclude? The fitness has increased in the experiment very rapidly initially with respect to time. Okay? And then there's a very slow increase. It's increasing, still it's increasing, but very slowly. And he fits his answer to this function, log of W bar, to be something like this. Some A and B are some constants, which he figures out by fitting the data. The data has been fitted to this function. So do you think any of this calculation has anything to do with that? Did we learn anything? This is the experimental data, and this is the fit to the data. This function is fitted to the data. and there was some calculation. Do you think there was anything in the calculation? Is there anything anywhere the calculation has a meaning? Or is it totally waste of time? What do you think? So what did we want? We said that the W bar increases exponentially or log W bar increases linearly with time, right? Do you see any log W bar increasing linearly with time? Yes? Yeah, very good. Where? Initial phase how? Yeah, from that function? Yeah, good. So here again, you wish you had maths course because Taylor series log of 1 plus x when x is small is equal to x. Okay? So therefore, this fit when bt is very small, that is times a short, log w bar goes as ABT, right? So it is increasing linearly at short times, okay? Whether or not this AB matches R and all is a separate question, okay? Then, okay, and so therefore you can fit etc. And it turns out this R, if you just take this one, it doesn't work. You take clonal interference into account, it sort of works. This is something like what I said yesterday also in the evening, that when this guys, Lurie and Delbruck, they calculated standard deviation by mean, I said, they get 0.2, theory gives 2 or something like that, right? It's sort of similar-ish, but not quite good. So here also level of agreement is okay, but not like, all points falling, theory and experiment falling on top of each other. There are a lot of fitting to be done, especially since many of the parameters here are not known yet. So the point here I want to make is the speed is real in the sense that you do see it in an experiment. Whether or not this matches well with the theory and all, it's an okay, sort of thing. If you started with the simplest case, it doesn't work at all. So the process we have teased out after doing this, calculation which I didn't show here, is that clonal interference is important if you want to at least get sort of ballpark figure. Okay, that's one lesson that we learned. Second, what's happening at large times? This behavior is very different. This is not linear. How much is it? You don't need Taylor series for this. Can you tell me? Bt is much larger than 1. How does log W bar go? Bt bar is like 100. Bt is larger than 100, let's say. Then, how is it increasing? Logarithmically. So log W bar is going like, Bt is very large, no, compared to 1. So I can neglect 1, okay? So this is going roughly like log t with some numbers, okay? So what have we learned? The speed of adaptation we have been calculating, and lots of people have been calculating. This is real in the sense of seeing in the experiment. There is a finite speed of adaptation, but this speed, it becomes 0 or the rate of growth goes log t, not t after some time. So what has happened? I did a very simple model, periodic selection. That doesn't work at all, okay? We did clonal interference, which I haven't shown you, but people have done the calculation, and it sort of works. So what is this log t? How do I capture that? I'll just tell you the answer here, because we are running out of time. You have to take epistasis into account. So we haven't talked about it at all. So epistasis is saying, so I said, one mutation comes, second mutation, irrespective of what is there in the background, it just fixes there. It's the same effect. It just multiplies 1 plus SB or 1 minus ST. That's not really true. When a new mutation comes, it depends upon the current genetic background. Okay? So that interaction we have not taken into account. That's called epistasis. Okay? So if you do a more full-fledged calculation, what it means is that this distribution, when first mutation has come and spread in the population, this distribution doesn't remain the same. It actually changes to something like this. So at least that's the assumption in the model. So once one mutation has fixed, my background has changed, the genetic background has changed. When the second one is coming, the distribution, the available mutations are different. The distribution is different. It has gone from blue to pink. So when you take care of that and redo the calculation, you get log T. So that's what the theory is giving you. So that's the value of the theory. Okay? So you fit the data. A and B don't match very well, but the fact that you can get a T and a log T by including these processes, namely clonal interference and epistasis, that's where the theory was useful in understanding this experiment. Okay? So the calculations are sort of simple. So if you understood yesterday's lecture and you're interested, I think you will be able to do it yourself. Okay? So I think I should stop now because there's a loo break now.